Welcome students. This is Dr. Sima Fernandez from St. Xavier's College, Mapsa, Goa. The course code is BOC 106. Course title is Cell Biology and Plant Biochemistry. Title of the unit is Cell and its Components. Module name is Other Cell Organelles 3. Outline of this course: structures of lysosomes, peroxisomes and glyoxysomes, functions of lysosomes, peroxisomes and glyoxysomes. Learning outcomes: describes the structure of lysosomes, peroxisomes and glyoxysomes, explains the function of lysosomes, peroxisomes and glyoxysomes. Let us see the other cell organelles 3: lysosomes, microbodies, peroxisomes and glyoxysomes. Introduction. Lysosomes are membrane-bound, dense granular structures containing hydrolytic enzymes responsible mainly for intracellular and extracellular digestion. The word lysosome, what does it mean? Lysosome is made up of two words: lyso, that is lysis meaning breakdown, and soma meaning body. It is formed from Golgi apparatus and occurs freely in the cytoplasm. It is commonly found in animal cells and only in some lower plant groups like slime molds and saprophytic fungi. In animals, it is found in almost all cells except in the RBCs. They are absent in bacteria. It occurs in most abundant numbers in cells related to enzymatic reactions such as liver cells, pancreatic cells, kidney cells, spleen cells, leukocytes, macrophages, etc. Now, this is the structure which shows the endoplasmic reticulum, that is a rough endoplasmic reticulum and smooth endoplasmic reticulum. Closer to it is the Golgi apparatus, and a Golgi apparatus is releasing the vesicle and the lysosome. So this structure is actually showing you that lysosome is formed from Golgi apparatus and it is released freely in the cytoplasm. Let us see the structure. They are mostly globular. If you see this structure, they are globular or granular in appearance. The structure shows the outer membrane, yes, and it shows the proteins and the enzymes. They are polymorphic. It is 0.2 to 0.5 micrometer in size. It is surrounded by a single lipoprotein membrane. So this membrane, what is marked in the structure, is made up of lipoprotein membrane, unique in composition because the membrane is made up of lipids and proteins. That's why it is called as lipoprotein membrane. The membrane contains highly glycosylated lysosomal associated membrane proteins called as LAMP and lysosomal integral membrane proteins LIMP. LAMPs and LIMPs form a coat on the inner surface of the membrane. Its membrane has a hydrogen proton pump which is responsible for maintaining pH conditions of the enzymes. Now, lysosomes are filled with hydrolases enzymes required for degradation of extra and intracellular material. It contains about 40 varieties of enzymes. Let us see the types of lysosomes: primary lysosomes and secondary lysosomes. Primary, it is small sac-like structure enclosing enzymes synthesized by the rough endoplasmic reticulum. So the primary lysosome is synthesized by rough endoplasmic reticulum. Secondary lysosome, it is formed by fusion of the primary lysosome with phagosomes. Primary lysosomes, it is simply called as storage granules storing enzymes. Whereas the secondary lysosomes, it contain engulfed material and enzymes. Materials are progressively digested. Let us see the functions of lysosomes. They are involved in the breakdown and intracellular digestion of macromolecules like carbohydrates, lipids, proteins, and nucleic acids. These are all macromolecules. It repairs cell membrane. They break down excess or worn out cell parts. They may be used to destroy invading viruses and bacteria. Let us see the microbodies. There are two types, peroxisomes and glyoxysomes. Introduction. Microbodies are single membrane cytoplasmic organelle of a more or less globular shape that consist of degradative enzymes. These have a central granular or crystalloid core containing some enzymes. They contain flavin-linked oxides and catalases for the hydrogen peroxides metabolism and also enzymes for fatty acid metabolism. Types of microbodies includes peroxisomes and glyoxysomes. Let us see the difference between peroxisomes and glyoxysomes. Peroxisomes, it is single membrane bound organelle, whereas glyoxysome has single membrane. Peroxisome, it is found in animal cells and in plant cells, whereas glyoxysomes found only in the plant cells, abundant in germinating seeds and filamentous fungi. Peroxisome, they contain catalases and oxidases, example D-amino oxidase and urate oxidase. Whereas glyoxysome, it contains enzymes of glyoxylate cycle besides catalases and oxidases. Peroxisome in plant cell, it remains associated with the endoplasmic reticulum, chloroplast and mitochondria. Whereas glyoxysomes, it remain intimately associated with lipid bodies, the spherosomes, and contain enzymes for fatty acid metabolism and gluconeogenesis. Peroxisomes, it is involved in photorespiration and it is formed by budding off from the endoplasmic reticulum. So the peroxisome is formed from endoplasmic reticulum, whereas glyoxysome, it is a special peroxisome. Let us see the structure of peroxisome. On the right hand side, you can see the structure of peroxisome, which shows the lipid bilayer, the plasma membrane, and the core is the crystalline core. Let us see in detail. It is variable in size and shape, but usually circular in cross section. Range from 0.2 to 1.5 micrometer in diameter. It consists of a single limiting membrane of lipid and protein molecules enclosing the granular matrix. Matrix consists of fibrils or a crystalloid structure containing enzymes. Let us see the structure of glyoxysome. On the right hand side, you see the structure of glyoxysome, which shows the outer membrane, inner matrix core, and inside the core. Let us see in detail. They resemble with peroxisomes. In morphology, it consists of single membrane enclosing the finely granular matrix. So if you see the structure, it is marked over there as matrix, but this matrix is finely granular matrix. In other words, it is a granular matrix. Its crystalloid core, the core is crystalloid, consists of dense rods of, can you see this core in the center, which is crystalloid, and it consists of dense rods of 6.0 micrometer in diameter. Let us see the functions of peroxisomes and glyoxysomes. Peroxisomes and glyoxysomes. Peroxisomes, it is involved in the formation and degradation of hydrogen peroxide by catalase. Glyoxysome, it is involved in the photorespiration and conversion of fats and carbohydrates. Peroxisome, they detoxify poisons of the cell, whereas glyoxysome catalysis of the formation of acetyl coenzyme A from fatty acids stored within the germinating seeds. Peroxisome, it is involved in the photorespiration. Let us see the functions of peroxisome. It forms amino acids through glycolate cycle. It is involved in the breakdown of very long chain fatty acids through beta oxidation. So, peroxisome structures are very, very important because it is involved in the breakdown of long chain fatty acids, that is called as beta oxidation. Peroxisomes in seeds are responsible for the conversion of stored fatty acids to carbohydrate. These are the references. Thank you.